timeline. Just now, we are going to start Dr. Banu Oral's presentation, one of the APMAS pre-recording. She's from Gebze Technical University, Turkey, and the title is Electrochemical Nucleic Acid Detection based on PNA probe. Dr. Banu Oral, do you hear me?
Yes. Hello.
Welcome again. Hello again.
Hi.
Hi. How are you?
Thanks.
If you are ready,
Okay,
I can start
share
your presentation.
Okay.
Hello everyone, I am Banu Oral. I am an assistant professor in Gebze Technical University, Institute of Biotechnology. Today I would like to present my previous study with the title of Electrochemical Nucleic Acid Detection based on PNA probes. Here is my outline. I would like to define briefly what's a biosensor and what type of electrochemical biosensors we used in the study and why we choose peptide nucleic acids as a probe. Briefly, I explain, and after that, what we did in the study, materials and methods, I explain, and after that, I'll give results on AC voltammetry measurements, and with conclusion, I will finish this presentation. What is a biosensor? A biosensor is an analytical device that combines a bioreceptor with a transducer to detect, analyze qualitatively and quantitatively. Here you can see the figure of biosensor components. We can classify electrochemical biosensors as bioreceptor. First one is biocatalytic sensors, the other one affinity biosensors. Here we use PNA as a probe, so we can call our biosensor device electrochemical affinity biosensors. As a bioreceptor, we choose peptide nucleic acids. Peptide nucleic acids are synthesized artificially. DNA have a deoxyribose sugar backbone, whereas PNA have N2-aminoacetylglycine backbone. So PNA contains no charge phosphate groups. Because of that reason, PNA-DNA hybridization stronger than DNA-DNA hybridization, why we choose peptide nucleic acids as a probe. Here is the performance of biosensors. We have already commercial biosensor devices. However, what we need to improve this biosensor performances. The most important performance are sensitivity, selectivity, and fast response. These are for all types of biosensors. We try to improve more sensitive, more selective, and very fast response. We try to detect infectious diseases and also the cancer cells at very early stage of the disease. And additionally, we try to produce point of care devices. These are patients can detect with these devices at home, not need any laboratory equipment. And for that reason, we try to additionally improve these biosensors to produce easy to use, cost effective or multiplex, and also the disposable ones we try to produce. So performance of biosensors are very important. Of course, to improve this performance of biosensors, the production process in each step, improvements are very effective at the final biosensor device. As the first step, electrode cleaning procedure is very important. We effectively immobilize PNA probe and ferrocene species on the surface. So electrode cleaning procedure very important on this part. Two types of cleaning respectively. First one is physical cleaning and the second one is chemical cleaning. Physical cleaning, we polished gold electrodes with alumina powders, and without powders we polish. And in chemical cleaning, hydrogen sulfide was used. Here 60 cycle, we obtained 60 cycles is the optimum cleaning we obtained from gold electrodes, and after that directly we placed into the immobilization solution to cleaning gold electrodes. Here we optimized the immobilization process. How to immobilize, two-stage immobilization process we used, and the first stage and first 10 minutes of this immobilization stage, temperature around 50 centigrade degree temperature was used. As a first stage of immobilization, we use ferrocene species as a redox species, and mercaptohexanol was mixed, and gold electrodes immersed to this ferrocene mercaptohexanol solution. Two different concentration was examined. One of them is 101 molar ferrocene mercaptohexanol, and the other one 120 ferrocene mercaptohexanol. Here we use mercaptohexanol to prevent non-specific absorption of complementary DNA hybridization. And again, I repeat, 50 centigrade degree was applied at the first 10 minutes and directly passed through the second stage of immobilization. Here we immobilize the PNA as 5 micromolar concentration on gold electrodes. You can see the figure or sketch of this immobilization process. And after the immobilization process, all measurements were done, actually AC voltammetry method. What we try to see, first after immobilization process we measured with AC voltammetry, and after that complementary DNA hybridization we applied to this mixed sums gold electrodes, and after hybridization process, we measure AC voltammetry again, and we saw the differences between the probe and complementary, after complementary DNA hybridization. Again, the 5 micromolar complementary single-strand DNA was used as a complementary DNA. All measurements was doing with AC voltammetry. 3 electrode system were used, and all measurements were measured in 10 millimolar PB buffer, pH 7, and the ionic strength was 19 millimolar. AC signal amplitude was 10 millivolt, and here is the potential scan range and scanning frequency range. We measured, and here are the results, what we measured. First, we compare the different two concentrations of ferrosanin mercaptahexanol immobilization on some surface, which one is more stable and which one is more stable. For that reason, we compare formal potential measurements to time and two electrodes simultaneously detected. And from these graphs, formal potential in graph A, 101 molar ferrosanin mercaptahexanol showed the same results. Decrease in the formal potential, but stable between two different electrodes. We calculate the surface coverage from the Ferguson species by using this equation. You can see in the slide. And after that, we plot the surface coverage versus time graphs. Again, the electrodes, simultaneously two electrodes, measurements. Here is the surface coverage decrease with time. However, stable results between two electrodes in graph A, 1 molar ferrosanin mercaptahexanol. So we choose this molarity and continue with PNA immobilization. Here are the AC voltammetry measurement results. First, after PNA immobilization, as a probe, the red plot, 3 repetitive measurements was taken or measured, the current versus potential graphs. After baseline correction, after complementary DNA hybridization, the current decrease and also the potential goes to negative. And as a result, we can say stable and reproducible measurements were obtained after complementary DNA hybridization by applying immobilization temperature around 50 centigrade degree and two-step immobilization procedure. It was determined small but stable negative change of formal potential after complementary DNA hybridization with high ionic strength concentration. This study shows that if PNA is organized with alkanitol sums effectively, highly sensitive and selective electrochemical detection can be accomplished to develop electronic biosensing devices. And thank you for listening. For questions, please send email to me. Thank you.
Hello again. Thank you, Doctor, for your nice and very interesting presentation. I think it's very interesting, one of the interesting research. Yes, I want to ask to all of our participants, do you have any questions to Dr. Oral? If you want to ask any questions, you can write your questions to chat part, or if you want to ask your questions by yourself, please write about you have a question to chat part. we can wait a few minutes. Yes, I think there's no question for now. Thank you, Doctor, again, for your nice presentation.
Thank you to you, and thank you for listening.
Thank you so much. And also, I want to say again, if any participant want to ask any question about this presentation, you can send an email to Dr. Banu Oral about her presentation. Yes.